All right. Hi, everyone. I know that some of you have said you might pop in a little bit later. I'm starting just a touch early because we do have quite a bit of material, some really great questions from you guys. And I wanted to clarify a couple of things to begin with here. So, the first thing is I do want you guys to be aware that you can use a cheat sheet. Okay. Your cheat sheet needs to be one side of one normal piece of paper, one side of a normal sheet of paper, handwritten, and it should include only things you can barely remember. It should just be bullet points or small amounts of information. I did post a video that I really like about the cheat sheets and how you should go about doing them a couple like last week sometime. So hopefully you've watched that. What I'd like you to do when you're written up your cheat sheet is send it to me in an email. So please send in an email. I'll send an announcement reminder as an attachment. This can just be a picture of the page. I want to see front and back, okay, to see that you've only written on one side before you take your test. Just be honest, and I'll ask you to show it again during the respondus. So show again during respondus. Environmental stuff, okay? Um, so that way I'm just making sure that you're being honest and doing what you should do. Cheat sheets really should be very minimal amount of information, and when you're studying, it should be something that just consistently slips your mind or something that you can use to trigger that memory. So as I'm going through the review today, I am going to point out some of the things that I think might make sense for a cheat sheet, and some of the things that might help you while working on studying for your exam. You do have a lab final due today. If you haven't taken that, please do. And then your final exam is going to be due by the end of the day on the 7th. So do make sure that you get that taken. You'll have 24 hours, just like a normal one. It opens on the 6th, and it'll be due by the end of the day on the 7th. You'll have an hour and 50 minutes. It is 37 questions. 2 of them are extra credit, 35 total questions for 175 points, and it is 15% of your total grade. So hopefully you have been studying and preparing for that, okay? So what I like to do when I review is I like to begin by just when I look at a unit, asking myself, what is the major concepts that we covered in that unit? And then I'll talk to the different questions you guys had. So for me, I wrote these like major concepts up first, and then I went through and built PowerPoint slides based on the questions that were presented. So for me, I say unit one is what is life, right? What is life? What does it mean to be alive? And generally, it's kind of more of the outside of what we actually think of as living or when we think about whole organisms. So we did talk about the characteristics of life in the scientific method. We also talked about chemistry, so chemical bonds, and then the properties of water. We definitely got into those macromolecules, which kind of tail off of chemistry. And then we talked about cells and organelles. And so none of these are really what we would consider a whole organism until we maybe get down to the cells. And even then, we dug into the organelles themselves. I will say that for each of your questions on the final, you do have a couple short answer, a couple matching, and maybe a fill in the blank, or you might have a short answer, two matching, two in the fill in the blank. It's roughly that. And they should be questions that you've seen, so you should be able to take them pretty quickly. Okay, so for unit one, the biggest thing we need to ask are what are the characteristics of life? And we know these characteristics. The first being organization, so like cells, right? Then we had response to stimuli. We have homeostasis. We have reproduction, which includes growth and development. We have adaptation, which includes evolution. And then we have metabolism, which is energy processing. So all of these things are required in order to be considered living or biotic. When we talked about ecosystems, we talked about the biotic versus abiotic factors. So biotic means you must have all 6. So one of the questions someone asked was, why aren't viruses alive? Which we did have on our first exam in Unit 1. And the biggest reason is that viruses don't have all of these. So viruses do not have cells, Okay. They don't actually do any sort of reproduction. They replicate inside of a cell. That's a little bit different. And they don't have their own metabolism. So viruses steal metabolism from the organisms that they've infected. And so we don't see viruses as alive based on the current characteristics of life. But those characteristics are always changing and developing. If you go and Google it, you'd find that there's more than um, just the six in some instances. And scientists are always having debates whether or not viruses are alive. Technically, because of what we are learning as the six characteristics of life, they are not considered living because they do not have all six characteristics. The same thing with, um, cars. Cars can respond to stimuli, especially those new self-driving cars, right? They have homeostasis. You have a signal that says, hey, dummy, your gas is getting low. They absolutely evolve and change over time. There's energy processing, but really cars don't reproduce and they don't have cells. And so our organization really does come down to cells at that point as well. So that should be a really quick recap. I think most of us understood that pretty well. And then we talk about the scientific method. Now, this looks complicated, but you guys did this all semester. All semester, you used the scientific method in labs to go through and test these labs. I asked you to make observations, write hypotheses. You were often asked to record your data and your results after following the steps of the experiment, and then sharing it with me by doing your lab workbooks is the final step of the scientific method. So if we were to write the steps in order, we would say observe, make an observation. Okay, that should be your first one. Observation, observe, ask a question. Okay. And again, remember, we do this on the regular. This is not something that we only do occasionally. We are making observations and hypotheses all of the time. So the next thing that you do is you would write a hypothesis. And in our level, I ask us to do if-then statements. So first we observe, then we write a hypothesis. Next, we would do an experiment, right? We would run an experiment with variables. And we know that those variables are the control, which is not changed, right? The independent, which is changed, and the dependent, which is the measured change. Okay, so that's our data that we collect. Okay, 4 would be your results. Okay, or conclusion. We can say results and conclusion because you can't draw a conclusion without results. You absolutely need to have that. And so they have on here analyze results and data, and it's a little more steps and a little bit more information. But this is still, I'm simplifying this beautiful image that's here. Okay. And last but not least, we would communicate. That final step is really important. If you don't communicate what you've found and you're not sharing what you've learned, we haven't really fulfilled the scientific method. Think about it if you find that there's a battery dead or batteries are missing and you talk to your partner or your kids or your mom or dad about it, that's going to be something that plays a role in making sure everybody understands what happened that the TV wouldn't turn on. Okay, so that's the really brief overview of chapter 1. I think the majority of questions that we had on unit 1 were on chapters 2 and 3. So chapter 2 was our chemistry of life. Okay, and the chemistry of life is unfortunately, it seems to be a snag for people. So we have to remember that when we look at cells, when we look at humans, we're made of cells. When we look at our cells are made of macromolecules. When we look at the macromolecules, those are made of chemical bonds. And so when we say we're looking at glucose and talking about cellular respiration, we're really looking at the way the chemical bonds are going to interact between the different elements that we see as naturally occurring. And especially in living organisms, we're concerned with the 5 big elements: carbon, hydrogen, oxygen, nitrogen, and phosphorus, which are why we talked about those in our biogeochemical cycles. So in our Chemistry of Life chapter, I introduced and talked to 3 different kinds of bonds that involve electrons. So if we remember, the electrons are the negatively charged subatomic particle that's found outside of the nucleus, where we would have the protons and neutrons in the nucleus of an atom. So we know that there are 3 different types of chemical bonds that we're interested in this class. If you cannot remember these, write them down, okay? Write down a shorthand note to yourself. I personally really like abbreviating. So nonpolar covalent bond, I would call the NPC, okay? And if you're familiar with any games, it's a non-playing character, and kind of that's what a nonpolar covalent bond is. It really is just content to do what it does. So nonpolar covalent bonds share an electron, which we can identify as an E negative, equally. There's no charge. And they're showing that in this image here with our Cl and Cl. So both of these are chlorine. Chlorine in itself is its own molecule. A chlorine bonded to a chlorine, neither one is bigger. So we're going to see a shared electrons right here, okay? And those electrons are just right smack dab in the middle between both of those chlorine nuclei, okay? And so we're going to see that that isn't going to be pulled one way or another stronger because they're equal here. So that is our first one. Let's talk then about the next type of chemical charge. Then we have polar covalent bonds. Okay, and I would do polar covalent bond, PCB. And if I know that nonpolar means no charge, I would assume, because biology is full of opposites, that polar means a charge. And so nonpolar covalent bonds would be no charge. Polar covalent bonds means they share electrons unequally and there is a charge. Okay, and that is illustrated in our example here, B, where we have, let me change the color here so that we have a slightly different color that shows up maybe a little bit better. Let's do this green. We have our polar covalent bond, and there's a hydrogen and a chlorine. The chlorine is much, much larger than the hydrogen. You can see that it has 1, 2, 3, 4, 5, 6 valence electrons on the outside on its own, plus 1 that's loose. So it's got 6 paired valence electrons and 1 that's loose. So we want 8 total on the outside. And so it's going to share that electron with hydrogen, but hydrogen only has 1 electron to share. And so hydrogen is going to be attracted to the chlorine, and what ends up happening is this hydrogen's electron gets pulled more towards the chlorine. We see a stronger pull towards the chlorine, and that means that we end up with a slightly negative charge and a slightly positive charge on the opposite end of that bond, with the hydrogen being slightly positive because its nucleus is slightly positive here, and the chlorine being slightly negative because it's pulling that negative charge closer to itself. So where we have our nonpolar covalent bonds being not charged, and this would be something like N2 or O2, right? Neither oxygen nor nitrogen is stronger than the other oxygen or nitrogen. We're going to see in our polar covalent bonds the very classical example being water, H2O. Okay, so water is going to be our really classic example, and that's going to come into play in our properties of water on the next slide, which was the next big question I had from you guys. And then our last kind of chemical bond that is dealing with electrons is the ionic bond. And this is the give an electron, receive an electron. Okay, and what this does, and this classic example of this is NaCl, and I like saying Na plus Cl minus because that gives me an idea that I'm looking at a charge. And what we're seeing here is that this sodium, okay, this sodium here has a extra electron, and it doesn't want that. It's got this free floating electron hanging out. It doesn't want that. So it's going to donate the electron to the chlorine. Let me see if yellow shows up a little bit better here. Orange maybe? Let's try orange. It's going to donate that electron to the chlorine. It's going to be like, nope, you take that electron. And chlorine, because remember, it has 1, 2, 3, 4, 5, 6, 7 valence electrons, it's going to be more than happy to pick that up. But now chlorine has one more electron than its protons, making it slightly negative, and sodium has one less electron than its protons, making it slightly positive. And this then results in a charge, and these are attracted to each other, but there's no sharing, okay? There's no sharing here. The attraction is because of the charge. Okay? And so so when we see this, we know that this is going to be something that it's closely associated to each other, but it is also very easy to break apart. Salt breaks down very easily when we put it into some sort of solution. That's why we have the oceans, and the oceans are salty. Okay? Now, that brings me to this idea of water, right? What is water, and why does that matter? So water is a little bit different. Water has that polar covalent bond, which, if we remember, is why we have this slightly negative oxygen and this slightly positive hydrogen. Here would be my shared electrons, right? I have those electrons being shared in the middle, and they get pulled more towards my oxygen. But this is one water molecule, okay? So that's one single water molecule. In this drawing, I have 3 water molecules. 1, 2, 3. Let me label them. 1, 2, 3. Water, because it has that slightly positive hydrogen and slightly negative oxygen from the polar covalent bond, has another phenomena that we see, which is another type of bond that we call hydrogen bonds. Okay? And a hydrogen bond is a weak attraction. And for our purposes, we would say between 2 or more water molecules. What that means then is that I end up with these beautiful dotted lines. Let me grab a different thing here. Let's try red. Okay. These dotted lines that we see between this oxygen of number 3, right, and this hydrogen of number 2 just means that this oxygen is attracted to this hydrogen. And when oxygen is attracted to hydrogen, there's a slight charge. It's kind of like our ionic bond, but there's no giving and taking of electrons. They're just having a slight attraction here. And then that will result in these connections between water molecules. These connections between water molecules are what give us the properties of water. So because of hydrogen bonds formed by polar covalent bonds, water has various properties, and I'm going to list those properties here. Cohesion, adhesion, surface tension. Okay, I'm going to make this a little bit bigger. I'm going to decrease my font size just a touch. Okay, surface tension. We have temperature resistance. Okay, and then things like density change when it's lighter as ice, or we have things, other various factors. Now, my favorite to talk about in terms of the polar covalent bond and the hydrogen bonds, because I think it makes the most sense to people, is the cohesion and adhesion. Cohesion, if we remember, means to stick to itself. So this is water sticking to itself. Well, this image on the side here with these polar covalent bonds and hydrogen bonds and showing all of this connection here is water sticking to itself. A drop of water forms because water sticks to itself. Okay. Adhesion, if you use an adhesive that's like a glue or a tape, and this is water sticking to something else. Well, that would be like if I had, let me get my thing up here. Okay. Awesome. Hello, Amanda. So I'm going to properties of water and chemistry a little bit. I already kind of ran through the characteristics of life and the scientific method, and then the little bit of the bonds, the nonpolar, polar, and ionic. We have a lot of really good questions for today. So if there's anything that comes up while I'm talking, feel free to raise your hand or just jump in, okay? Otherwise, just welcome aboard. So if we talk about these polar covalent bonds being cohesion, This is that water sticking to itself. If I had a leaf on the side here, and this leaf was slightly negative, which is very likely to happen, this little hydrogen is going to stick to my leaf. This little hydrogen is going to stick to my leaf. This is a really bad leaf, but there's my leaf, okay? So when I get a drop of water on a leaf, what's happening is the water's positive hydrogens are sticking to the negative leaf's surface, and the water charges within the water molecules in that drop of water are sticking to themselves. And so we end up having water sticking where we wouldn't see that with other things. Like salt has a charge, but if I stick salt onto a leaf and I just put it on the leaf and there's no moisture, the salt falls right off. Okay. If I spill water on my countertop, the water's going to form little, it's going to form a pool. Then as I clean it up, it kind of sticks to the surface a little bit. When I spill salt on my surface, I can just pick it up. The salt doesn't stick even though it has a charge. And that's because the salt doesn't have the hydrogen bonds that are forming that stickiness, whereas the water does. So when we talk about the properties of water, and there was a big question on the Unit 2 exam about this, asking what kind of bond forms between an oxygen and two hydrogens. Well, that's your polar covalent because they're sharing electrons. And then what kind of bond forms between different oxygens and hydrogens of multiple water molecules? Those are your hydrogen bonds. And we only see the properties of water being sticky or being resistant to temperature, having surface tension, any of those things that we've talked about, the universal solvent, because of the polar covalent bond that creates this positive and negative situation that then creates this attraction between multiple molecules, as well as just kind of some loose hanging, like positive and negative charges that can interact. Okay? Any questions about what I've covered so far? If not, that is okay. Because I'm going to move on to the last big set of questions from chapters 3 in Unit 1 was about the macromolecules. Now, there are no tables on the final exam. So for anyone watching this later, there are no tables on the final. I don't ask you to build me a table on cellular respiration and photosynthesis. I don't ask you to build me a table on transcription and translation, or even on your macromolecules. But it might be useful to draw a short, small little table on your cheat sheet if it's something that you struggle with. Macromolecules we covered in basically every unit for the entire semester. And so they do play a role in comprehensive sorts of questions, such as, tell me about the macromolecules involved in protein synthesis and how we build proteins. So that's kind of a short, quick example. I don't want to give you the actual examples, but it is meant to ask you to think about some of those things a little bit more in depth in terms of how they relate to things to like the biogeochemical cycles. So I do want to build out a really, really quick macromolecule table because this was requested. So we have carbohydrates, we have lipids, we have proteins, and we have nucleic acids. Those are the 4 macromolecules or biomolecules that we talked about this semester. Now, all of these have monomers and polymers, except for our lipids. Lipids are a little funky. So if we talk about carbohydrates, the monomer is the monosaccharide. Okay, the polymer of carbohydrates, let me see if I can, yep, there we go, is a polysaccharide, which we all know and we're very, I think, comfortable with the idea of mono versus poly when we're talking about that base word there. Saccharide just means sugar, so you've got your simple sugars and your complex sugars. This could be glucose for your simple sugar or starches for your complex sugars. When we ask what elements are in carbohydrates, what I'm asking is which kinds of molecules are we finding, which kinds of elements on that periodic table. So for carbohydrates, the name tells us it's got carbon, hydrogen, and oxygen because carbohydrates. Lipids don't really have a true monomer and polymer. because they're so varied in what they are and what they do. This includes things like hormones and waxes and phospholipids, as well as fats and oils and things. But they still have carbon, hydrogen, and oxygen. The note here is these are nonpolar and these are polar. So if we go back to our chemical bonds, we know that a nonpolar covalent bond has no charge because the electrons are being shared equally between different molecules and there's no overall net charge, but a polar has a charge. And so we know that carbohydrates are going to have a charge and are going to interact with water, whereas lipids are nonpolar and they do not interact with water. When I mix oil and water, it doesn't mix. Okay? Proteins are amino acids, which we talked about in protein synthesis. We use those amino acid monomers to build polypeptides, which is our polymer of proteins. And this includes a new element called nitrogen. And this is why nitrogen is so important when we talk about biogeochemical cycles, because nitrogen is a limiting factor for plants to build their own proteins. They need that nitrogen in order to do that. And they build their own amino acids. That's why we can get so many good amino acids from plants. And then in nucleic acids, our monomer are nucleotides, and our polymer are nucleic acids like DNA and RNA. And we now include carbon, hydrogen, oxygen, nitrogen, and phosphate. Phosphate also plays a huge role in those biogeochemical cycles and what we're doing to disrupt them by, adding fertilizer or different sorts of pollution. And we also need phosphate for ATP because ATP is adenosine triphosphate. So I would say that you should make note of these elements and where they are in the macromolecules and what role they play in biogeochemical cycling. I also had a question about hydrolysis and dehydration synthesis. So I just wanted to kind of recap on that. When we're talking about hydrolysis and dehydration synthesis, what we're doing is we're either taking monomers and turning them into polymers by removing water. So our first example is dehydration synthesis. And remember, if you can break the words down, you know what it means because you know that synthesize is to make and dehydration is to remove water. So we are making by removing water. So we're adding a glucose to make a starch, for instance. Or if I want to break that starch down into glucose so that I can use it in cellular respiration and get energy from it, I have to add water into it, which is hydrolysis. So hydro meaning water and lysis meaning to split. So we add water to split a polymer into monomers. And then we can use those monomers and break them down further and rearrange those molecules. But the very simple part of this is when we talk about things like protein synthesis and we're talking about how we're building amino acid sequences into polypeptides, what we're doing is we are taking and we are going through dehydration synthesis. Or when we talk about DNA being duplicated through mitosis and meiosis, we are taking nucleotides and building nucleic acids using dehydration synthesis. But when you eat something, if you eat a strawberry, you don't take that strawberry's genes, the DNA in that strawberry, and just shove it right into your genetic material. You break it down from its nucleic acids into its nucleotides. And so then you're doing hydrolysis. So these are just ways of recycling these monomers and polymers back and forth depending on what you need them for. I do really like to think about it in terms of Legos because you could take Legos apart and build them into something else. Now there are some specialized pieces, but occasionally if you get one of those basic sets, you can build basically anything out of it and you would just retake those different ones and arrange them into a new way to make something new, okay? And I think that's it for Unit 1. If you're not comfortable with cells and organelles, make sure that you can match those organelles to what they do, as well as make sure that you recognize what all cells have in common, which, just real briefly, would be the cell membrane, the cytoplasm, and genetic material. Okay? Not a nucleus, genetic material. My seated students have told me nucleus 6 times this week, and I'm like, no, that's not right. All right. Apologize. Unit 2 was focusing on metabolism. So if I were to look at Unit 2 as a whole and say, how is this related to biology? How is this related to the characteristics of life? I would say that it's metabolism. We're looking at energy processing. And for me, I ask, what can I do to summarize each chapter here? Chapter 5 is kind of an introduction to metabolism. So we learn about some of the factors that make metabolism possible, like enzymes, concentration gradients, diffusion, and osmosis. Chapter 6, we learned about photosynthesis. And chapter 7, we learned about cellular respiration. So for the most part, people's questions based on this unit were how to make the tables, how to know the inputs and outputs. Again, that's not on the final. There are no tables on the final. I use these more as jumping off points for some of those comprehensive questions. And so I'll talk to that after I kind of run through a couple of the things that I did have some questions on that were specific. And I thought that's probably good to cover for the cumulative final. So the first one being enzymes. And there are a couple of things that came up as questions for enzymes. This one, just, go through enzymes again. 2 was go through this graph again and figure out how to read it. And 3 was talking about the types of inhibition. And I saw the inhibitor question quite a bit. So if we remember, enzymes are proteins that catalyze reactions. They speed up reactions. And so I have this image here that shows a line with a very high energy spike. So if we read this side, it says energy up. Very high energy spike that reads without enzyme. And that is the activation energy required without the enzyme. I also have a figure underneath that with a smaller hump that shows with enzyme, and it's about half the required activation energy, or maybe even a third, to start that process with the enzyme than without the enzyme. And so that is showing us that it's easier, it's faster, it works more efficiently when an enzyme is present. If you see a large line and it's saying this is energy and it's a really high amount of energy, chances are that means there's no enzyme present. My favorite kind of analogy for this is this idea that I could pound a nail into a piece of wood by myself with my hands, my arms and hammer with a hammer and nail. I could do it. Now, I'm not a carpenter, so it would take me a little bit of time and effort, and I would probably be sore about it later. But I could do the same job with a nail gun, and it would be faster and more efficient, and it might be more accurate. I don't know about you guys, but I always end up bending my nails when I try to hit them with the hammer, but the nail gun just puts it in nice and straight and where it should be. And so the enzyme is like having a tool that does the job better and faster than if you do it by yourself. You can still do it by yourself, but having the enzyme present makes it more effective. So you guys could study by yourself, but having a cumulative review video makes it more effective where you're asking me those questions. So that was one of the big questions. The other one was about the inhibitors. Now, enzymes are controlled in a variety of ways. One of the biggest one being that enzymes are specific to certain substrates. So if I have an enzyme, it has an active site that is only going to fit a certain substrate. And so I have this image up top here where we see the active site, and it matches to these substrates. They go in and bond. The enzyme has some of the hydrolysis effects, and so we'll break those bonds between these two parts of a polymer, and will release the two products that can be worked on further, and then the enzyme does its job again. This is not wasted. It keeps going through that process. Now, if I don't want the enzymes to do that, I'm a cell and I'm trying to build up a concentration of this substrate for some reason, which happens a lot, I might stop my enzyme from functioning by using competitive inhibition, where my inhibitor is already in that spot. And so the substrate tries to bind and gets bounced off. Okay. This is like if a cat is using a dog's bed and the dog looks there all sad because it's not going to bounce the cat from its bed. Okay. It can't use it anymore. It's not available to it. That's the same thing with the substrate. The competitive inhibitor blocks the active site. So it just blocks the active site completely, and that enzyme cannot bind to its substrate. Non-competitive inhibition, though, is when I have a non-competitive inhibitor bind to an alternate site. Okay, see, this is an alternate site, and it changes the shape of the active site. And then the substrate can't bind because there's not enough space for it. This is absolutely parking at the base at the mall when base is off, right? You go to park and someone's 4 feet over the line and you can't park there because that parking space is partially eaten up by something else. Or someone left a shopping cart in the parking space. So you can't pull in there because someone put the shopping cart in the parking space. It's changing the functionality of that active site. Now, cells use both of these as well as a lot of other types of inhibition, but these are the ones that I really want you guys to know. So make sure that you kind of, if you can't remember it, draw a little picture. Pictures are worth 1,000 words on a cheat sheet because instead of reading a big full sentence, I can look at a picture and go, oh, right. Competitive inhibition, active site, got it. If I write out a full sentence, then I sometimes have to read that full sentence, and if I miss a word, I might mess it up. The more that you can simplify your notes on your cheat sheet, the better that cheat sheet will function for you. So that was enzymes. And then there were some questions about concentration gradients. And there weren't any questions about diffusion and osmosis. And I hope we're feeling pretty confident about that, especially after doing the egg lab. It's one of my favorite labs. I love that lab. But when we're talking about concentration gradients, what we're really talking about is when you have too much of something on one side of a barrier. Now, nature hates concentration gradients, but we use them in our cells all of the time to control homeostasis. If my cell just lost water whenever it wanted to, or if my cell did not have a way to hold on to its glucose and then go through that process of cellular respiration, well, it takes a little bit of time, then it is more of a problem, right? I want my cell wants to hold on to the things that are inside of it. I want a barrier that says not everything can move in and out. Honestly, the best way for this is just think about a fence, right? Think about a chicken fence. It's not going to let the chickens out. It's not going to let the predators in, but insects go in and out. Maybe small reptiles go in and out, like those lizards, but they're not affecting the main goal, which is keeping the chickens separate from the predators. So your cells work like a giant fence. And there are gates in that fence that sometimes allow things to move more effectively. But often what we see is things moving through diffusion and through osmosis. Diffusion is solutes moving from high to low. Osmosis is solvents, most of the time water, moving from high to low. And in osmosis, we talked about the 3 osmotic environments, and I am a big old fan of drawing these and just having them sketched out with the note. So if it's hypotonic, here's my cell in a hypotonic solution. Water is just going to move into that cell, and my cell is going to burst. Bloop, 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 burst. Hypertonic, here's my cell again. Water is going to leave my cell just like crazy. All these arrows just mean water, and then that means my shell is going to shrivel, and it's not going to look like a cell anymore. Isotonic, everything is happy, and we have even in and even out. If you just write down those things and draw those little pictures, usually that kind of helps people grasp it. You guys did really good on that one, though. I wasn't surprised to not see any questions from anybody on it because you guys seem to grasp that concept really well in this online section. So kudos, all the power to you. What you should see then is on this exam, because it is covering enzymes and concentration gradients and photosynthesis and cellular respiration, the majority of the Unit 2 questions I pull are going to be mostly enzymes and concentration gradients, because cellular respiration and photosynthesis, there's a question in Unit 2 on that, obviously, for the exam. But they came up over and over and over and over and over again all semester. And so they are inherently a comprehensive question where you are going to tie cellular respiration and photosynthesis into other things we've learned about, like fitness and evolution. So let me just talk about concentration gradients in photosynthesis and cellular respiration, as well as just briefly talk about photosynthesis and cellular respiration. Again, there's no tables. So if you can look at something and say, oh, cellular respiration is the mitochondria, you're golden. If you can look at something and say glycolysis is cellular respiration, you're golden. If you say the chloroplast is photosynthesis, you're fine. If you try to tell me that photosynthesis is in plants and cellular respiration is in animals, I'm going to be a little bit cranky because we've talked about how cellular respiration is in everything that's alive. That happened last semester at the end of the year, and I was like, guys, what happened? So keep that in mind. You guys get to learn from all of their mistakes. So when we talk about concentration gradients generating ATP and NADPH, this happens in both cellular respiration and photosynthesis. This was a question on the test. It was, I think, an extra credit question, but this came up more than once when people were reviewing and studying. The very simple answer to what do concentration gradients do in cellular respiration and photosynthesis is that they generate ATP. Okay, both processes need to make cellular energy before it can do anything else. So this is the first step of photosynthesis, is to make ATP. The light comes in, we see this as the light, and that light-dependent reaction. Hydrogen ions are pushed into the thylakoid membrane, so we see that we're getting a concentration of hydrogen ions. We're going to see a lot of hydrogen ions down here, and they are looking for a way out because, again, nature doesn't like concentration gradients. And so these hydrogen ions move through something called ATP synthase and generate ATP. In cellular respiration, it's the last step. It's the ETC. It's the third step. And what happens is, again, we are pushing hydrogens into a space that's creating a concentration gradient of high hydrogens, and those are going to try to move to a low hydrogen environment, which requires them to once again move through the enzyme called ATP synthase and generate ATP. Both have to do that. If I want to trap cellular energy as sugars and I'm using solar energy as my starting point, I can't just go solar energy to sugar. I have to go solar energy to cellular energy to sugar. And so that's why we see ATP being generated in photosynthesis. And then the opposite is true in cellular respiration because these are, in fact, opposites of each other. Okay? Questions yet? Are you good, Amanda? You're my only person on, so I'm going to ask.
I'm good so far.
Okay. I feel like you were one of the people that asked one of those questions, so I was like, let me just make sure.
I did.
Yeah. Does it make sense, though? They're just making ATP. That's the whole point of a concentration gradient.
Yes. It makes more sense, like, going back through it.
Yes. It's always harder when you've taken the test and then you look at it again. But once we've talked about it, usually people go, oh, yeah, that's right. It makes more sense. It clicks. So I'm glad to hear it. All right. The other thing I'm going to tell everyone that they should just write down on their cheat sheet, like the covalent bonds, like the bond definitions, write down the equation for photosynthesis and cellular respiration, but write it down like it's in this image here, where it's the 6CO2 plus 6H2O going into glucose and oxygen with photosynthesis on the top and solar energy coming in, and then the opposite arrow with cellular respiration and chemical energy coming out. This will 100% make your life better, because if you get into the test and I ask you something about the equation, you might have it down, but we've all been in those cases where we brain fart and we get into the test and it's just gone, brain gone. I have no idea what that equation is. I blanked. I don't know. Have it just written down. It just makes it easier. And honestly, it shows us by writing it out this way that they are opposites of each other and that they play a really big role in balancing the carbon and water cycles and the oxygen cycles. These carbon and water cycles are really important, and we're disrupting them as humans. And so that's more of what we might see as a comprehensive question. The other thing that's usually helpful for people is if we write down or draw a little image. Some people, this image helps them a lot. Sketch it out rather than writing down all of the nitty gritty details. That's going to be really helpful. When you're studying for this part, make sure you can define these words and make sure you can tell me, is this word photosynthesis or is this identify, so like define, and then tell me photo or cell, photosynthesis or cellular respiration. If you can look and say, oh, right, ATP, both, right? Plants and algae, photosynthesis. The chloroplast, photosynthesis. Mitochondria, cellular respiration. You'll be really good to go for that part of the exam. Okay. I know everyone was worried about the tables, not on the final. Really good study tool, good note-taking tool if you feel like making a table on your cheat sheet, but absolutely not required to have you pull all that information out of your head again. I tested you on that once. We did it really, really well. I'm just looking to see if you can recall and match that stuff now. That's my testing at this point. Okay. So if everyone feels that I didn't do photosynthesis and cellular respiration justice, you can go watch the Unit 2 review, but also just remember it's more about this. So I wrote down an example comprehensive question, and I pulled up this image that's kind of showing what's happening. So an example comprehensive question would be something like, what is the relationship of cellular respiration, photosynthesis, and the carbon cycle? And how are humans disrupting the natural balance of these processes? Now, this shouldn't seem like a crazy question because we talked about this. We know that cellular respiration and photosynthesis are opposites and they balance the carbon cycle. We know that the carbon cycle is being disrupted because of combustion and fossil fuel use, as well as burning of plants and cutting down of rainforests and killing of animals and ocean acidification. And so when we talk about how those two processes would normally balance out the carbon cycle, it's not working that way because we are disrupting it. And it's just because if we go back to the equation, you could pull the equation out and say, look, it's 6 CO2 and 6 C. So that carbon is being trapped as glucose. But if we're removing plants that can take up that carbon dioxide, it can't be trapped as glucose. And so it's hanging out in the atmosphere and it's getting absorbed by the oceans and it's leading to ocean acidification. There's not really a right answer for a good portion of this. What I'm looking for, I mean, there's a right answer, but what I'm looking for is to see if you can bring these things we've talked about all semester and just think about it a little bit critically. Critical thinking is one of the major objectives of biology. And tell me, I see this connection and I understand why we had to learn about cellular respiration and photosynthesis to fully appreciate ecosystems and the balances that exist in there. Hopefully that sort of thing shouldn't throw anybody off too much. Another one that becomes a really big question is, why do we breathe oxygen? And my 110, my seated students would tell you this. I ask this question almost every single class. And every single class they tell me it's to live. Right. But why do we breathe oxygen to live? And how does that relate to fitness? And it really is just the idea that we breathe to provide O2 for cellular respiration. That's it. That's why we breathe, which provides us with energy. Energy is fitness, because without energy, you cannot make babies, right? That's the sort of thing that I'm looking for. For the cumulative or comprehensive questions, there's 5 of them. 3 of them are 5 points, 2 of them are 10 points. And so a 5-point question might be something like this. It's not going to be either one of these questions exactly, but if you can give me a good solid answer like that, you don't have to write me a book. You have to just show me that you understand why all of these things are connected, and that's just biology, right? We talk about fitness, we talk about community interactions related to fitness, we talk about evolution related to fitness, and at the end of the day, that's all making babies, and babies take energy. And I feel like I say this 700 times and I need a t-shirt that says it, but that's the sort of question you might see more related to photosynthesis and cellular respiration than the nitty-gritty details on photosynthesis and cellular respiration. Okay? Does that make sense? Any questions on that one?
No questions.
Awesome. You get to be my sounding board. It's beautiful. Usually I talk to myself during these, and it's I feel a little bit like a crazy person sometimes. All right, Unit 3, we would talk about as reproduction, and I'm going to tell you right now that if you haven't reviewed your vocabulary, go review your vocabulary. Pull out those old flashcards, dust them off, make sure that you understand the major vocabulary for Unit 3. But this is mitosis and meiosis, and again, there were a lot of questions about identifying the differences between mitosis and meiosis. You've had a test on that and you've had labs on that. I'm not going to show you an image and ask you to identify it, but I might ask you to tell me the differences. And so I built out a table to help us kind of run through the similarities and differences of mitosis and meiosis. Now, when I ask for a comparison, you got to give me both parts of the whole. I gave the example yesterday of if I asked you to compare apples to oranges and you simply describe apples to me, that tells me nothing about the orange. So you have to give me both. If I say compare mitosis and meiosis, and you say mitosis is asexual reproduction, which is great, that's true, you have to tell me then that meiosis is sexual reproduction. And you need to be very clear on what is what. Please don't make me guess. If I have to guess, I'm going to mark it wrong because at this point in the semester, we should all know that I want a very clear delineation of your answers. If you go in and you tell me that mitosis is 2 diploid cells, and you can even 2 identical diploid cells, right? Then meiosis is 4 unique haploid cells. I was telling my seated class yesterday, go for the low-hanging fruit, okay? It doesn't have to be the most out there answer. I want you to just tell me what you recognize, what you know, right? So this is used for growth and repair, right? This is used to make babies. That's a low-hanging fruit. If I say that this goes through PMAT once and this goes through PMAT twice, easy. If I say that mitosis, I'm trying to think if there's anything else crazy that I was like, let's talk to this. I don't think so. But I might put on here a couple of things that were unique about meiosis. And this is I might add to my cheat sheet if I were someone who maybe missed this question on the Unit 3 exam. Crossing over, independent assortment, and fertilization. Now, fertilization, I'm going to put a note here, happens after meiosis, and it doesn't happen for all meiotic events. Otherwise, women would not have periods. We would just be constantly pregnant, so that is not going to happen. But fertilization, independent assortment, and crossing over give us unique individuals, okay? And I have another slide about that, so I'm going to come back to that. But if you are someone who forgets that these are a thing, put them on your cheat sheet. As far as what they have in common, they both go through interphase, right? They both make new cells. They have a parent cell that's diploid, right? You could even put cytokinesis, but is cytokinesis part of mitosis or meiosis? No, it's its own thing. Okay, so you had questions more than once in the quizzes and on the exams asking you to compare these two processes. Expect to see that again on the final. Now, You also might see something about why we see organisms using asexual reproduction when sexual reproduction is also a possibility. And this ties into Unit 5 a little bit. So when we talk about the type of reproduction organisms use, we think about it in terms of benefits and detriments. So asexual reproduction is generally faster, okay? It's easier because there's no need to attract a mate. Okay, there's less energy, and it guarantees offspring with your DNA. That's kind of a big deal. It guarantees fitness. However, they're all identical, which means no resistance. Okay, and that's usually the biggest downfall. And you cannot do it if more complex. So humans, right, we're very complex. We have cephalization, we have segmentation, we have specialized organ systems. We can't do asexual reproduction to make a copy of ourselves. That's not going to work because we're too complex to do that. So we do see asexual reproduction being the choice in a lot of organisms that are going to be in varied environments. So if this is going to be a stable environment, we might see asexual reproduction, but we can look at that in terms of sexual then, and this is another one of those comparison questions where it really is kind of low-hanging fruit, where the benefit of asexual is it's faster, this one is slower. Does it take energy? Yes. Is it harder? You got to attract a mate, right? More energy. Okay, you can still have offspring, but it's not always a guarantee, especially if we talk about female choice and some other weird things that you would get in a higher level biology. And then this is going to be in a variable environment. Okay, obviously this energy part is a huge, huge part because that ties back to cellular respiration. In the detriments here is that it's lots of energy, costly, and finding a mate, right? And choosing a mate can be very, very complicated. But we actually see variable environment. We actually see varied offspring. So where we see our detriments there, it's mostly the energy. Everything, for the most part, can do sexual reproduction. A lot of organisms just choose to do asexual reproduction because it's better. The biggest detriment of sexual is that it's energy. So let me just take that off of here. These actually should be detriments, not. There we go. Okay. So that would be something, I saw that come up a couple of times because that comes into play again when we talk about the diversity of life in Unit 5. So keep that in mind. Briefly, let's talk about the cell cycle. Cytokinesis is its own step. And remember that we have 3 phases of interphase, but the cell spends most of its time in interphase. Don't spend too much time on this. It's very short. Also remember, what's the disease of the cell cycle called? That's cancer. I cannot stress enough how much you need to just look at your vocab for this unit. And then remember the difference between animal and plant cell cytokinesis. So in animal cells, we have a circle, and we're going to get this cleavage furrow. Cleavage furrow. Okay, and in plant cells, because there's a wall of cellulose, we end up with a cell plate. Because I cannot split something that has a hard wall by pinching it in two. I have to literally cut it in half. So that, I think, was the only thing for the cell cycle. There were some questions on alleles versus genes and a couple of questions about genetics. So when we talk about alleles versus genes, remember, alleles are alternate forms of a gene. The gene is what codes for the phenotype. And then your genotype is the genetic code, what's actually physically in your body. And the phenotype is the physical traits. So we know that individuals can have the same genotype. They could be homozygous dominant for a trait. And if they have that genotype, they're going to have the homozygous dominant phenotype. But if an individual is also heterozygous, that means they have a dominant allele, and that is going to make their phenotype also dominant. And so I have this image here that's showing attached earlobes and free earlobes. And because this person inherited one of each, they're heterozygous. They have a capital E because that's the dominant trait, which means that this individual's phenotype is going to be the dominant trait for free earlobes. Again, I think this particular sort of question series I got from people goes back to vocab. Go back and look at that vocab list. Go back and try and answer those. And remember that genotype and phenotype play a role in evolution. And that was more of a comprehensive question on the Unit 4 exam. Make sure you have a look at that as well. This one is that sexual selection, how does it make us unique, and how does that relate to Mendel's law? This was like 90% of the questions that I had from people. So let's go through it real quick here, okay? One, we know that in meiosis, we are looking at homologous pairs. So we are looking at the chromosomes that came from mom and the chromosomes that came from dad. And you may not have the same information from mom and dad. Now, those chromosomes are going to line up in a homologous pair in metaphase 1, which is your dead giveaway that we're in meiosis, if we see pairs like this. And they're going to separate out into different gametes. Now, if they line up and they don't do any sort of crossing over or independent assortment, we are going to see that 2 of the gametes produced are going to have the same genetic information on each side. So this first one here, right? This is big R little y. This is big R little y, right? So dominant and recessive. This is little r big Y, little r big Y. And that's just how they inherited it straight down from the top here. Because we see here's our dominant Y, here's our recessive R. And that just went right down that line. That went right down that line. But if we have independent assortment occur, so if we have where my chromosome pairs after they've lined up split out randomly, I might see here RY, 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 RY. And even though these 2 gametes are still the same, that is showing us how we're starting to get that mix up through that independent assortment. And we can have chromosomes lined up at the end that do not match exactly what we had in the parents. Now, if we add in a swap of genetic material here, okay, and then maybe here, so maybe these swapped genetic material. Well, now I have brand new chromosomes that are lining up, and that's crossing over. Crossing over is this swapping of genetic material where I would have, let me do my draw, and let's do a marker here. So if I have a chromosome like this, and then I have one that's like this green area in the middle here is going to swap segments. We're going to see 2 new chromatids then, 2 new chromosome combinations that have not existed. So crossing over exchanges alleles and genetic material. Independent assortment shuffles them up into different gametes on their own without any contribution or control from another set. And then random fertilization is because not every single sperm and every single egg are going to come together to make a baby. It's going to always be random. And so that is what gives us our genetically unique individuals through sexual reproduction. And this relates to Mendel's laws because when we look at the Punnett squares, right, Mendel said, okay, cool, we're seeing this mixing up of parents' genetic information in each generation afterwards. We know that we are going to have unique individuals because of these 3 processes that are the same as what Mendel observed in his piece. Okay, I feel like that was another one of the questions maybe you had, Amanda. Does that make sense?
Yes, it was more of the independent assortment portion of it.
Yeah. And I think that was where everyone got stuck up. I think people get crossing over. They're like, yeah, they exchange segments. Got it. It's the independent assortments. That's why I pulled this picture in particular too.
Right.
Okay, awesome. Keep that in mind. And if you haven't studied for that one, go look at that. And that's for everybody. Go look at that question on the past test. Hint, hint, nudge, nudge. Okay. I don't want anyone to pull answers though and put them on their cheat sheet. If you do that, I'll have to give you a 0 and it'll be a bad day. So we're not going to do that. But go study that material. So there was a question about Punnett squares. I just wanted to talk to this idea of generations. So our first generation is our parent generation or pure generation. And then this first cross, this is my first cross. These are my first hybrids. Okay. This is my F1 generation. And if I say you're going to cross the F1 generation, this is literally all you have to cross because everybody is heterozygous. And so you would have the female gametes being dominant and recessive, the male sperm being dominant and recessive. And so your F2 generation is always crossing your heterozygous individuals in this sort of true breeding lineage. I know some people got a little confused with that. It's just asking you, this is your pure generation, your first generation. We're crossing it, and then this is my second generation of hybrids. So if I let them self-fertilize, which remember peas like to do, that's what Mendel did, you find a divvy up here. And I wanted to talk then to the genotype and phenotypic ratios. So in my F1, my genotype is 100% homozygous, or sorry, heterozygous, right? They're going to have a capital T and a lowercase t. And in my F2, my genotype is 1. I went to phenotype in my head too fast. To 2 to 1. So in my F2, we see down here they have it written out that I have an individual who's homozygous dominant, 2 individuals who are heterozygous, and 1 individual who is homozygous recessive. My phenotypic ratio, my F1, the phenotype is 100% tall, or the dominant trait. Okay, in my F2, we have 3 that are tall with the dominant trait, and 1 is short, the recessive trait. And this is showing us once again that your phenotype doesn't have to be the same, doesn't have to have the same genotype. So if you are recessive, you will always have the same phenotype and genotype. So if you have blue eyes, you have the recessive trait for blue eyes, which means your genotype is recessive. But if you have brown eyes, I don't know if you are recessive or dominant because you only need 1 dominant allele to give you that dominant trait. I think I wouldn't give you any sort of dihybrid Punnett squares. It would be a very straightforward generation like this where I would give you purebred individuals and maybe ask you to do a Punnett square like that. If you are struggling with Punnett squares, let me know, but also just go watch the Amoeba Sisters video. I cannot stress enough how beautiful a job they do explaining that. In ways that I wish I could animate everything for you guys. I just can't. I wish I could. I'm not that talented. I did want to also talk to pedigrees really quick because this could show up on the exam as well. Remember, square is male, circle is female, right? Shaded is affected. Non-shaded is not affected. And then half shaded would be a carrier. If you see half shaded, it means that they're heterozygous. In this instance, this is a pedigree particularly for color blindness. And we know that this is sex linked in particular because males are more likely to be affected. If we look In our first generation, we have a male who's affected. So this male is XY, and he's going to have that small n. Their daughters are not affected because mom doesn't have that trait and she's not a carrier. So we know that these daughters are carriers, or this daughter is also a carrier. We just happen to know. And then the son inherited for both of these, all these sons, they inherit the dominant trait from mom because X comes from mom. Okay. We know that this has to be sex linked because we have 1, 2, 3, 4, 5, 6 males affected, right? And we have one female in the fourth generation who's affected. And we have 1, 2, 3, 4, 5, 6, 7 carrier females. Okay. Females are more likely to be carriers because they have 2 X chromosomes. When you have an X and a Y, you make sperm with X and you make sperm with Y. When you have an X and an X, you make egg with X, you make egg with X. You don't have that. So if these two come together, you get a boy. If these two come together, you get a girl. And so we know in this instance that these males only inherited one X, and they get that X from mom, which is why in this first generation, we do not see any males who are affected, but in future generations, we do. I think that's what I wanted to cover on that one. And then I just briefly wanted to talk to 2 of the comparative Mendel's variations. There's the incomplete dominance and codominance. I love the flower example. If I were to cross a red and a white flower and I get pink, that is incomplete dominance because red plus white makes pink. But if I were to cross this red and white flower and get stripes, that's codominance because neither one is masking the other. They're both equally observed. Codominance in humans, the classic example is blood type, and a lot of people got that one wrong on this exam. I am AB blood type. My husband is O, so he's recessive. I have the dominant trait for A and the dominant trait for B, which means that I can give a baby the A blood type or the B blood type, but I cannot give them a recessive blood type. I do not carry the O. There's no carrier here. My husband has the O, and so he is going to give them the recessive allele, but our babies would be either A or B, never O, because that is just not a possibility for the simple level that we're sitting here. Go take blood bank and you'll maybe learn different things if you join the MLT program. But for our purposes, that question on that test, I cannot have a baby with O blood type. Even if I had a baby with someone who wasn't my husband, it wouldn't matter because I do not have the recessive trait. I also talked a little bit about pleiotropy and polygenic inheritance, and I really liked this image here. So one gene affecting multiple traits is pleiotropy. And a great example of that is sickle cell anemia, where you're going to end up with blood clots and vascular damage and organ damage and potentially other painful side effects. But it's because one gene is wrong. Where polygenic explains how we have some families that will end up having a child who is a giant even though everyone else is an average height. And that's because you have multiple genes that affect height. And so they inherited all of the dominant genes and just ended up on this far end scale. Polygenic inheritance is very common in humans. Skin color, hair color, technically eye color is often polygenic as well. And then there was a question about environmental factors. And there are ways that environmental factors can affect your physical expression and affect your genes. Don't worry about that for the exam, but if you're curious about it, Google epistasis and epigenetics, and there's just a huge area of research that's showing that if your great grandmother grew up during the Depression and struggled with food security, and went through periods of starvation, you have a higher risk of obesity because of the changes that occurred in her alleles from that difficulty at that point in her life. It is really, really cool, but that is some high level genetics that I don't know that we even fully understand yet. And it's absolutely mind-blowing, and I think we're learning more and more, obviously, with the better technology we get, which is helping us appreciate that. So my big thing for Unit 3 for you guys is please, please, please go review your vocabulary. That particular unit, it wasn't the worst exam. I think the only thing that's hard on it is if I ask you a question using the vocab, you have to know what that word means in order to answer it correctly. And if you don't know what that word means, when we come to the final exam, there's not a whole lot I can do to help you. So pull out those flashcards, recap them, look back at the past exam matching, make sure you can look for definitions to things. Okay, I think we're on to Unit 4 then. And right on time, good. I try to do the first 3 units in the first hour and the second 3 units in the second hour. So, all right, buckle up. You ready? Let's talk about Unit 4. Unit 4, protein synthesis. Again, no tables being built, but we will talk about why that's important. And then we did a lot of evolution. Protein synthesis gets jumped into evolution because it's the link between the genetics aspect, where we talk about genotype and phenotype, and the natural selection and evolution aspect, where natural selection acts on phenotypes and changes gene pools over time. So I had a question about the differences in DNA and RNA. And I think the big differences we need to be aware of is that DNA is double-stranded, RNA is single-stranded. DNA has thymine, RNA has uracil. DNA stays in the nucleus, RNA moves. I think if we can keep that straight, that will help us do really well with matching or identifying protein synthesis. So that's why I have this next part here. And I have just a brief table because, again, tables are really, really fantastic ways to organize your thoughts. And putting tables on cheat sheets is a great way to keep things in a very easy manner for your eye and brain to find when you need that safety net. So transcription always comes first because C is before L. Transcription occurs in the nucleus. DNA makes messenger RNA. What's involved? DNA, promoter, gene, RNA polymerase, mRNA, nucleotides, nucleus. When I say what's involved, what I'm looking for is if I am trying to find a correct answer to say this belongs to transcription, I want my key words. I want the words that are going to give it away for me and make my brain go, right. So if I say that transcription is the process of converting DNA into RNA that leaves the nucleus, that tells me that I'm looking at transcription. If I ask you to match transcription and translation, which is what I did on the exam, and I say, hey, this occurs in the nucleus, you should be able to tell me that it's transcription. If I say that nucleotides are added to mRNA, you should be able to tell me that's transcription. If I say that genotype, the DNA is the genotype and is coded for messenger RNA, you should be able to tell me that's transcription. If you look back at that chapter, that unit, all of these are your key words for when we talk about the initiation, elongation, and termination process. Translation occurs at the ribosome, and second, in the cytoplasm or on the rough ER. That immediately separates these two things for me. If I say, oh, this happens at the ribosome, you should be able to tell me that it's translation. If I say this is in the nucleus, you should be able to tell me that's transcription. That also tells me that I'm taking messenger RNA and I am making proteins. That's my big goal, because the whole thing of protein synthesis, that central dogma of biology, is DNA to RNA to protein. That's the big deal. And this is conserved across organisms. This is hugely important for all of our living organisms. Bacteria do it, archaea do it, all of the eukaryotes. So everything we talk about in Unit 5, all of the domains, all of the kingdoms, everybody does protein synthesis because proteins do the work, okay? So what's involved? mRNA, right? tRNA, which is your transfer RNA. We put transfer RNA, and then ribosomal RNA, which is rRNA. I might say something about amino acids. I might say something about a polypeptide. But at the end of the day, that's what I look for. That's what my keywords would be. That would be the thing that gave it away. If I said a growing polypeptide, that is not going to happen in the nucleus. Proteins are too big. They got to get out of that nucleus and go somewhere else. So we're going to see that happening in translation. If I say transfer RNA brings an amino acid, absolutely translation. If I say mRNA, tRNA, and rRNA form an RNA complex, absolutely translation. We should be looking for those keywords. And then it's not so much that, like if we know those keywords, that's important. But the big thing is, why is this so important to understand? Why do we care about protein synthesis? One, it is conserved across living things. Okay. Two, proteins do all the work. They are your phenotype. You have proteins that do all of the work. You have proteins that say you have blue eyes. You don't think about it that way, but that is what it is. And this is the source of mutations. If your DNA has an error in it, that source of mutation changes the phenotype and might lead to evolution. That is why this is so incredibly important. Now, I won't be asking you to use a chart to identify an amino acid, a codon sequence as an amino acid. That was in your lab. There's no table, okay? But these important parts, why we talk about this, is really going to be more of a comprehensive sort of question. And you saw the question about that on the Unit 4 exam. So I would go have a look at that and make sure that you understand how protein synthesis leads to evolution. Okay. And then I had a quick question, or a question that should be pretty quick, about these mechanisms of evolution. Again, I always look for keywords. And if I can't remember the keywords, these are the things that are going on my cheat sheet. So mutation, change in gene sequence, right? Cool. It could be good, bad, or neutral. So let's put a plus, a minus, and a 0, because it could be good, bad, or neutral. Okay. Natural selection is a pressure on survival or fitness. Always it's a pressure. It's a pressure. That is your keyword there. And if you read anything and it sounds like some sort of pressure, it sounds like a predation event. I'm trying to avoid a predator. Well, that is a stress. That is a pressure that is acting on that animal. So natural selection is always a pressure. Genetic drift is always a chance survival of a natural disaster, usually. Once again, keywords here. So if I think about genetic drift, I think it's just a chance. It's chance or it's random. A fire comes through. It doesn't matter if I'm blending into my environment. If I stand still during a fire, I'm going to die. So natural selection has no play there. Remember, the founder effect is a new population in a new area. And the bottleneck effect is large die-off, loss of genes. So if you think about endangered species, if we go through and we kill them all, or down to a very few amount, when we talk about our loss of biodiversity, if we take and remove those genes from the system, they are gone forever. They cannot come back. We cannot put them back. So that's why things like extinction events in Australia are so problematic, because we cannot put those organisms back. And if we even have surviving individuals, That's it. That's all there is. And they don't have the same genes as every other organism that ever lived in that population. We know that populations as a whole have a very unique array of genes, unless we've had something happen that removes a large portion of those genes. Gene flow, okay, is the movement or migration of individuals. So genes moving back and forth through situations, moving from one environment to another environment. You take your genes with you. So if I move to, well, I mean, humans are a great example of this, right? We move around all of the time, and we meet people of different cultures, and they have different genetic information than us. We are mixing up the gene pool. So this mixes up the gene pool. Okay? That is microevolution. Any questions for Unit 4? That was all I had for Unit 4 as far as questions were concerned.
I think I was pretty good on Unit 4.
Okay. I think most people were too. That one felt pretty good, I think. Unit 5. No tables. You can't use your tables. I know it's like awful, but honestly, it's going to be very straightforward for this unit, okay? Unit 5, we're going to talk about that diversity of life. So Chapter 17 was microorganisms. Then we did plants and fungi and animals. One of the things I wanted to hit on that came up on different units was this idea of domains and kingdoms. So we have 3 domains, bacteria, archaea, and eukarya, and that is the most inclusive. Domains are everything that's a eukaryote. You're a plant, animal, fungi, or protist. You are in the domain eukarya, which means we share at least some common traits that lead to a common ancestor. Within eukarya, we have 4 kingdoms: plants, protists, fungi, and animals. And then there was questions about binomial nomenclature and how we get species name. And this is the least inclusive. So we have an example here. Kingdom Animalia includes anything that's an animal: jellyfish, spiders, wolves, bears, skunks, crocodiles, birds, bats, everybody. Chordates include only the organisms with a backbone, or those 4 traits of chordata, not technically a backbone, but in this image, they're only showing the backbone. Mammals include organisms that have hair and fur. Carnivora includes organisms that eat meat. Canidae is the family, so this is talking about our dog-like organisms. Genus is Canis, that is including all the dogs. It removes foxes. Foxes are a different genus. And then our species of Canis lupus is our wolf. So you can see the wolf is included in Kingdom Animalia, but so are everything else. But when we get down to the species name, we're talking about a single type of organism. So that's why we do these classifications. We're really trying to narrow down where these organisms fit within these boxes so that we can really just better explain evolutionary relationships and have a better understanding of how they fit into their ecosystems. This does lead me to the key traits of major groups, and this is the sort of thing that I would ask you to do on the final. So for instance, if I said something has a peptidoglycan cell wall, that should give you bacteria immediately in your head. If I said something has 3 common shapes, and they're the spiral, sphere, and rod, immediately should give you bacteria in your head, not like physically bacteria in your head. That's bad. Don't do that. Okay. But that should kind of give it away. If I ask for any key trait of archaea, I'm going to say extremophiles. And this is the sort of thing that you might see. I might say organisms with a peptidoglycan cell wall. I would expect you to be able to tell me that that's bacteria. You know, organisms found in extreme environments. Expect you to be telling me that that's archaea. Okay. People asked about protists. Protists are a catch-all. This means that their key trait is that they are not a plant, animal, or fungi, but they are a eukaryote. Okay. This includes your algae and slime molds. algae, slime mold, and amoebas, for instance. You could also put examples here. So archaea, our example would be things like methanogens or thermophiles, thermoacidophiles. Bacteria, I think we're pretty good with bacteria, but this would be things like E. coli, Listeria, Salmonella. Okay. Protists are often unicellular, and they're usually small. Okay. And they might be mixotrophic, heterotrophic, or autotrophic. So they might feed themselves, eat other things, or do both. Plants, pretty straightforward, I think, for plants. They live on land. They are multicellular. They include moss, ferns, conifers, and flowering plants. They have vascular tissue. They photosynthesize. They're autotrophs. Okay, I'm literally thinking through the PowerPoint and like those first couple of slides where I said, here's what makes a plant a plant. That's the sort of thing that if you're writing something down for Unit 5, that's what you would want to put down, okay? Animals are multicellular. They move with neurons and muscles, okay? They have sexual reproduction required at some point in their life, and they have internal digestion as heterotrophs, which makes them different from fungi. Fungi have absorptive digestion as heterotrophs. They are mycelia or yeast, so they can be multicellular mycelia or single-celled yeast. And they have a mushroom for reproduction. They are really important decomposers. Okay, so that's the sort of thing that I would make sure that I knew. And then if I said something like something has feathers and lays eggs, we should all know that they're birds and birds are classified as reptiles. If I say something is furry and produces milk, we should say mammals. Well, has mammary glands that produce milk. If I say something is got thin skin and lays its eggs in water because it will dry out, then that would be amphibians. So nothing too crazy. Honestly, this is the biggest part. So make sure you know these ones. And then some of these little details that did come up. So one of the questions that I ended up throwing out on the Unit 5 exam, which we should all look at, is what do organisms have to overcome to live on land? And living on land is very difficult as compared to living in water. And I talked about it in terms of plants and animals. So let's look at it in terms of plants and animals. Okay. Now, when we talk about the difficulty of land, gas exchange is a problem, and gas exchange has to do with drying out. Okay, so how do plants overcome gas exchange? They have stomata that open and close for gas exchange. Animals will do lungs. Internal lungs are actually a huge advantage to living on land, and we start to see those well-developed lungs in our reptiles, or maintaining moisture. So things like snails and slugs are going to stay wet, right? If they stay wet, then they don't dry out, then they can do gas exchange effectively, okay? When we talk about gravity, it really depends, but for plants, that's your vascular system, so the xylem and phloem allow them to maintain themselves in gravity and stay upright. Let me make sure I make this clear here, gas exchange. So plants, animals, plants. For animals, skeletons. Absolutely. That could be the hydrostatic skeleton like we see in earthworms. It could be the bony skeleton we see in vertebrates, but having a skeleton with muscle attachment is going to be what allows us to handle that gravity pressure. Reproduction, again, is akin to drying out. Okay, we want to make sure that we're not drying out our babies. Plants, these are going to be spores, naked seeds with a seed coat, or covered seeds with fruit. And animals, they either lay their eggs in water, like amphibians, or they have an amniotic egg, which contains water, like our reptiles and mammals. And then, last but not least, UV radiation would be in plants, they have a cuticle, and in animals, we start to see things like staying in shade in our amphibians, right? If you have a worm or a frog that's out in the sun all day, especially here in North Carolina, that's going to be a very crispy little critter. That's not really good for it. Or scales, feathers, and fur, right? That helps us prevent UV radiation and sunburn. So I did want to just highlight those a little bit better. We deal with these difficulties in both plants and animals, and we find different ways to have the same solution. So this is an analogous trait. We don't see it the same. Like the cuticle of plants is not the same as the cuticle in insects or your finger cuticles, but they do play the same sort of role. So they are for protection. Your cuticles and your fingers are for protection of your nail bed. The cuticle in plant protects it from sunburn. So we have protection there. Okay. Briefly, why are birds reptiles? I pulled up pictures because I think pictures tell us everything. If we look at the beak of this turtle and the beak of this bird, they are not that different. They are made of the same things. They have the same structures. That is a homologous trait, meaning study of the same. Their feet are not that different. They're covered in scales and they have the same sort of bones, also a homologous trait. I think the scales are the biggest giveaway. Their scales have the same overlapping texture as bird feathers. They're made of the same materials called keratin. So that is also why birds are reptiles. And then the biggest thing is their eggs. If we look, eggs by birds have a hard shell, but turtles and snakes, their eggs are very similar. They're going to have a thick, leathery shell. These are all amniotic organisms, and that amniotic egg is really important to being able to live on land and seeing evolution happening in this group of organisms. Flight is secondary to thermoregulation, so it is strongly believed that birds first developed feathers when they were their dinosaur ancestors in response to changing temperatures, the atmosphere getting colder, and flight came second. So we do see that evolving later. Birds are endothermic. They do maintain their own internal body temperature. But other than that, they share a lot of common traits to reptiles, and current analysis from the fossil record and DNA analysis suggests that they are a very closely related group of organisms. So they should all be classified as reptiles. We have our avians, which are your birds, and then your non-avian reptiles, your turtles, lizards, snakes, and crocodilians. I know that was a question for a couple of people. And then the last question came up, and this is cephalization. So cephalization is the concentration of a brain and specialized organs. So if we think about our senses, we think about our 5 major senses. We have sight, sound, smell, scent, and then touch, which kind of isn't really one. Mr. Jackson, when he teaches A&P, says balance instead, which is better because balance is a sense. And all of those are concentrated in our head. This also is why we don't see asexual reproduction as organisms get more complex with their cephalization, that head, because how do I make sure that my brain gets split in 2 equally? You only have so much space, especially with a cranium protecting your brain. You can't add extra brain matter that would be divided into two separate organisms like we would see in asexual reproduction. So keep in mind that cephalization is a concentration of specialized senses in the head, and that is the brain. And that's true for any of our animals that have a brain. It is in one area. We have a brain. Plants don't have brains, and so plants can do asexual reproduction very easily. Fungi don't have brains. They can do asexual reproduction. Protists don't have brains. We don't see this nervous system until we see animals, which is why sexual reproduction is required for animals at some point, and why when we see more complex animals, we don't get asexual reproduction, unlike we could see in plants where they just put up little shoots of themselves and it's fine. Okay, I think that's one of the other big ones there. Anything for Unit 5 before I finish up with Unit 6?
I think I'm good.
Cool. I know it's like you're learning the whole semester over again in a sitting, but hopefully all of this is sounding familiar to everybody who's watching the
Right.
recording later. All right. Unit 6 is ecology. You guys did really well on this test. My biggest thing for this one was, what was the one that got me? Keystone species. I think we need to just re-clarify what the difference is between keystone species. So community interactions, I put this up here because there were some questions about that. There are 4. There is competition, which is negative, negative, and remember that plays a role in fitness. Consumption, which is positive, negative, again in fitness. Commensalism, which is positive, 0, and then mutualism, which is plus, plus. These all have to do with fitness. They all have to do with cellular respiration. So they all have to do with the carbon cycle. Maybe think about that in terms of a comprehensive sort of question. Okay. They all also play a big role. They are a pressure for evolution. You just put that on there. Someone asked me to give an example of when a interaction, I think what threw people off is I asked about when one of the example interactions from the matching could become something else. And I didn't mean for it to become that kind of confusing. So what I want to just clarify is that if I have 2 organisms competing for a food source, there are no winners. Okay, even if I have someone win, there are no winners. So competition is pretty cut and dry, except for when it's not. So one of my favorite examples of this, and let me see if I can find a picture of it. So badger and coyote out in the Midwest, okay? Badgers and coyotes are not friends. They compete for the same food source. So why are these 2 guys hanging out together? There has been recent documentation that in times of abundance, when there is a lot of prairie dogs available, there's a lot of food available, these 2 organisms don't compete anymore. They gear up and work together. And the coyote will find the prairie dog because they're taller. They will show the badger to where the prairie dog hole is. The badger will start digging because they are much better diggers than the prairie dogs, the diggers than the coyotes. The coyotes will find the back door that the prairie dogs would normally use to escape the badger. This only happens when there's a lot of food. When there's not a lot of food, it's every predator for itself. When that badger is scaring those prairie dogs towards that back hole, the coyote is there grabbing prairie dogs and killing them, and the prairie dogs are scared and run back into the badger's face and get eaten. Both of them get to eat, okay? Some of the examples that I gave you that some people caught on to were things like cows eating grass. That is absolutely consumption. The cow is eating the grass. But if the cow is leaving a fertilizer behind, that increases the grass's ability to grow, then it's also mutualism because the cow gets food and the grass gets nutrients. If we talk about Spanish moss growing on a tree, that's commensalism. Unless the Spanish moss starts blocking the leaves and affecting the tree's ability to photosynthesize, then it's competition. So don't that's not going to be on the final in that way, but I will ask you about community interactions and how they might be pressures for evolution and relating that back to some of these other big concepts here. This is one of those kind of big questions that would show up as a comprehensive sort of question. I did have a question about trophic levels. Remember, we start with our plants. Those are our producers. They bring in solar energy through photosynthesis. Okay, remember photosynthesis here. Okay. We are transforming that solar energy into cellular energy, and this is ATP. And we can say that about 10% of available energy as ATP is transferred, and the rest is lost as heat. So when we get to our top level consumers, there's not enough energy to go higher because 0.001% would not make it worth feeding at this level. Instead, it would be better to feed at one of the lower levels. So we lose energy as heat through cellular respiration. Okay, so food webs are a way of showing not just this straight line of trophic feeding like we see in the pyramid, but showing us all of the interconnectedness. So this is our food web from the exam. Seaweed is our producer, and so is phytoplankton. These are our producers. Crab then is a primary consumer, squid would be secondary, elephant, seal, and penguin are tertiary, and killer whale is quaternary. Krill would be primary, blue whale is secondary. Krill primary, fish would be secondary, seagull tertiary, leopard seal quaternary. If we do, right? Yep, and then also 1, 2, yeah, so then zooplankton is 1, fish is 2, leopard seal is also tertiary because we can fulfill more than one trophic level in this cascade. Now, your killer whale is the apex predator, not because no one else will eat a killer whale. I guarantee a leopard seal would probably eat a baby killer whale if it could. It's not about who eats it, it's about this level of energy. The energy you would have to put into catching and consuming that predator is not worth it. And so we actually would see organisms choosing and evolving to feed at lower trophic levels than we would see them trying to eat a killer whale. The blue whale is way bigger than a killer whale, okay? Absolutely, that blue whale is not going to eat the killer whale because that is eating krill. In fact, killer whales will totally eat blue whales. They'll eat baby blue whales, okay? When we talk about food webs, all we do is count the numbers of steps in each of these chains that make up the web away from the producer. So if you can identify the producer, you should be golden. There are no labeled decomposers in here, but crabs and seagulls are 1,000% decomposers. They are part of that decomposition food chain. There is not a food web on the final, but understanding how these trophic levels play a role in cellular respiration and photosynthesis, and therefore community interaction and evolution, would probably be helpful for a comprehensive question. Okay. And then keystone versus exotic versus invasive. This actually wasn't really asked of me. I think maybe one person asked. This is something I noticed. Okay. Keystone species increase biodiversity by being present. Exotic species reduce biodiversity. Nope, exotic species are just neutral. They're just introduced and don't spread. They don't compete. Invasive species reduce biodiversity by competing with native species. And examples, I mean, some people told me they couldn't remember North Carolina examples and gave me other ones, whatever, okay? Examples of invasive species are everywhere. Chinese holly, Chinese privet, Japanese honeysuckle, wisteria, kudzu. Those are just plants off the top of my head. Fire ants, pythons, nutria, feral hogs, spotted lanternflies. The giant apple snails are from Africa. Like there's a lot. And when these invasive species get introduced, what they end up doing to the food web is if I introduced an invasive species that now was eating all of the squid are gone. No more squid. Bye. That invasive species ate them all. Then we're going to lose the elephant seal. Then we're going to lose the penguin. We might have declines in the leopard seal. We might have declines in the killer whale. That is the really big problem with an invasive species. Okay. Last three things here real quick. So biodiversity and conservation. Woodland bee was more diverse on the exam. Okay. Woodland bee was more diverse not because it had more trees, but because it had a more even distribution of trees. So if I go in here again with my drawing, right, or here they have A, B, C, and D. These are my 4 types of trees. A, B, C, and D. I have A, right? I have B, I have D, and then over here I have C. Both ones have 4 species. Okay. But I don't think anyone would look at the second community and say, oh yeah, that one's great. This is being dominated by a single species, whereas this one is more diverse. Okay. We want biodiversity because biodiversity increases the direct and indirect benefits to humans through biogeochemical cycling, through clean air, clean water, food, just recreation. I'm a big fan of going out to areas that are different and unique. And if we can do things to preserve it, that's great. Everything that we do, drop in the bucket. It's all about your own karma and your feelings about what you do. I'm not saying that you need to 100% like this is exactly what needs to happen in order for my life to occur. And to be better and to do things for the environment. But, maybe try not to use those plastic bags to put your fruit in. Get one of the reusable ones and take it with you, right? Those little clear plastic bags. Maybe try to eat more locally as much as you can, or make sure that you're recycling properly. And just that little bit makes a difference. And if we are planting things in our own yards, do some research and plant some native things, and that's going to increase biodiversity and conservation. Conservation is really helping the environment for future generations. It's not about what we're going to reap from that, it's about what's going to be left for people in the future. So make sure that you are kind of tracking that. I think everyone for the most part got that, but if you're reading those questions, make sure you're answering all parts of those questions. Some people gave me 1 answer and I asked for 3. Make sure that you're filling all of that in on the final. I had a question about succession events, and I thought this was just the best picture I could give you. Succession events leave nothing left alive, and you have to start from scratch. Your paper didn't save anything, and you have to start all of your research over again. Secondary, you have something. It saved an old rough draft, and so you have something to work off of. Things are still alive. Burning is okay. It's a secondary, and after a week or 2, this is going to be green again. We won't see this lava field be green again until after the lava is done and cooled, and then after all of this volcanic rock has broken down. So primary succession takes longer and is more, you start from scratch, nothing's left alive. But secondary succession is faster, and there are things left alive. It's just removing, it's a good part of the nutrient cycle, to be honest. We are releasing some of these dead and decaying trees. We're just breaking down the carbon and nutrients faster and making it available to them. High level fires that are really intense are not good, but that's why we do a lot of controlled burns here in North Carolina and Onslow County. It's actually really beneficial, not just to prevent forest fires, but also for our environment and ecosystems as a whole. Okay. And then biogeochemical cycles. This one, You're not going to have any direct questions on a biogeochemical cycle. You will have a comprehensive question relating this to macromolecules, cellular respiration, and fitness. So think about biogeochemicals in that. But the ones we talked about phosphorus, we talked about nitrogen, and we talked about carbon. Well, carbon is in all 4 macromolecules, right? Nitrogen is in proteins and nucleic acids, and phosphorus is in, whoops, I could spell, phosphorus is in nucleic acids and plays a big role in that ATP cycle. I think that's, there's one thing back in cellular respiration, real quick. Fermentation, anaerobic, anaerobic respiration. Aerobic respiration requires oxygen, anaerobic doesn't. That gives us fermentation. Fermentation is done by yeast. It does not produce yeast. And fermentation produces various food parts that we like to eat, things that are tangy or sour. So like sour cream, uh, I was going to say bacteria, but that's not it because I'm thinking lactobacillus. Lactobacillus breaks down yogurt, so yogurt, or like beer and wine and cheese and bread. Okay. Phew, I know that's a lot. Thank you for sitting through this with me, Amanda. I appreciate it. Is there anything that didn't get to?
I don't think so.
Okay. That was my goal. My goal was to try to touch on everything. And hopefully, if you or anyone else who's watching the recording has questions, your exam is due on the 7th. So you have the whole day of the 7th, which is Tuesday. I think I'm opening it on the 6th. So you'll have that whole day to take it. If you're studying over the weekend, just send me an email. I'm more than happy to answer questions as they pop up.
I only have a question on the cheat sheet of
Yeah.
is there any kind of boundaries or rules on those?
I'm sorry, say that again.
Is there any kind of I guess how much? I know it's the one sheet.
Yeah, so it's one side of a normal sheet of paper. That is my requirement, and it has to be handwritten. You can put as much or as little information on it as you want, so long as you are not writing down answers from the past tests.
Sweet.
Okay. If you're just literally writing down answers from the past tests, we're going to have a big problem, and that goes for everybody. I personally, when I do my cheat sheets like this, I split it into 6 sections, and they don't necessarily have to be equal, but I would do a unit 1 section, a unit 2 section, and so on. And that way, we have a rough idea of what each unit covered. If I go, oh, right, characteristics of life, that was way in the beginning of the semester, I don't have to go searching through the whole thing. I can just go back to unit 1 and say, all right, here it is. That's my biggest recommendation would be to have it in 6. And keep it clean and clear. If you have it full up jumbled, you're going to not find what you need on it. I have had some students write the most insane cheat sheets, and then they're like, this was really hard to use. I'm like, right, it's meant to be a safety net if you forget a vocab word, right? If for some reason you cannot remember the steps of the scientific method in your head for whatever reason, you write those down. If you can't remember the equations, you write those down. That's more of what that's for. It's there to provide you with a safety net, because I know it's a lot of information. I know I'm not crazy. I understand, but it is, you know, still just a tool, and so you don't want it to be too full, or you won't find it useful for that final. But it's just one side, and I want everyone to take a picture and send it to me, and don't modify it after that. I also want you to show it during your Respondus environmental check. To just make sure that if, you should not be writing down answers from the past test. Even though I told you I pull past test questions, that is really inappropriate. You should just be studying. If that was the case, I'd just let you open your past tests, but that's not what we're doing today, okay? Does that make sense?
Yes.
Okay. Anything else?
I don't think so.
Okay. Well, and you're the only one here, so you get the final say.
Right.
I'll post this, and just as always, you guys send me an email if you need me. You guys are great. You've been doing such a really fantastic job this semester. Really proud of the work you've put out. So I'm not overly concerned. I know it's a scary thing to think about a big cumulative final. Most of the time, people do right around where they've been all semester. So if you've been sitting at your A all semester, you'll probably get an A on the final. That's just usually what it looks like. And I did apply the extra credit from the spring break and pre-lecture. So you've seen that. Your grade should reflect minus the lab final, exactly what should be in there for your grade right now. So cool. Well, if anything comes up, let me know. Otherwise, thank you, and I hope you have a great day.
Mhm.